Students, today we study secretory type of tissues, secretory structures in plants. Topic belongs to plant anatomy. Secretory structures and special types of tissues. The main function of these structures the release of substance that have a special physiologic function like release of enzymes, hormones, excretion, separation of product eliminated from metabolism. Secretion is used to include excretion. Role of many of these substances is not known. They may be found in the same container. Secretion may remain in the cell, producing it as droplets. Types of secretory structures found in plants we divided in two categories. External secretory structures and internal secretory structures. First we study external trichomes and glands, nectaries, osmophores, hydatodes. The second type is secretory cells, secretory spaces, latticifers. In first part we study external secretory structures. First one is trichomes and glands. Trichomes simpler than glands, although no sharp distinction exists between glandular hairs and glands. Trichomes are contains anti-herbivore secretions. Glands also like salt glands mechanism for the plant to get rid of excessive salt absorbed from the environment. Function of plants living in salt marshes and salt infiltrated soils in coastal marine areas. Glands of insectivorous plants secrete digestive juice. These are also known as glands. In example, we saw the glandular hair of Artica dioica, also known as a stinging plant, stinging nettle. This is the salt gland of Tamarix ephyla. Insectivorous plants. Structure of secretory type of cells have dense protoplast and large nucleoles. Mechanism for release may release secretion in between wall and cuticle. Eventually cuticle bursts or not are but individual cells are severed after release of secretion. Special mechanism like tip breaks off, contents histamine escape into wound. In these exams, in these pics we saw this type of secretion. Glandular trichrome, glandular hair of Artica dioica. These are the next type of nectaries. May occur on flowers or vegetative parts, extra flower nectaries, known as secretory tissue, restricted to the epidermal layer or may include subsurface layers also. Having dense cytoplasm, may be papillate, are closely packed, have thin walls. The diagram shows that these types of nectaries, various types of nectaries. Nectar is derived from phloem. Nectar excreted through cell wall and rupture cuticles. or through stomatas, they are not able to close and open. Osmophores, next type of secretory structures. Osmophores scent of the flowers produced by essential oil originates from epidermis of perianth from osmophores, special type of glands. These structures found in Aristolochiaceae, Araceae. Floral part differentiated as osmophores may assume the form of flaps, bushes, or cilia. May be distinguished by using neutral red. Some methods for locate to osmophores. Secreted tissues of osmophores. Hydrathodes, a structure that discharges water from the interior of the leaf to the surface. Water contains dissolved salts, sugars, and other organic substances. Eliminates water through the terminal trachea. In contact with epitheme, may have no epitheme and water moves through ordinary mesophyll. The basic purpose of hydrathode to release water. In this pic we saw guttation, the exudation of water droplet. The diagram shows the hydathode's internal structure. This is the first part of secretory structures. In next part, we study external secretory structures. In the second part, we study internal secretory structures.